Hej allihop och välkomna hit till våran studio och ska vi köra en liten webcast här igen. Det var ett tag sedan Mats, eller hur? Det var ett halvår sedan som vi höll på med det här.
Ja, tiden går, men vi är väldigt glada att Martin har velat ställa upp på den här intervjun. Han deltar på videokonferens från Oslo och det ska bli jätteroligt att få prata om honom om en del av de sakerna som har hänt här på slutet.
Welcome to our studio from Oslo, Martin. We're very glad to have you here. And I hope that we can hear you. If you just say a few words like hello.
Yeah, so happy to participate again. So I think it was a long time since last time.
Yeah.
But obviously due to the, how do you say, strange times that we're in, that's also the reason why I'm working currently from Oslo. So that's probably the main reason why we didn't touch base in between.
Right.
Okay.
So we have a few questions for you, and I think Mats, you take charge of this, and I'll see if I can follow up on anything or help. But please go ahead.
Thank you. Okay, Martin, we will start you off with a couple of softball questions and ask you about some of the exciting stuff that's come out lately, and then later on in the interview we'll talk about some other topics as well. So starting out with your most recent press releases about the abstracts, and specifically starting with BT001. So we saw the abstract ahead of the conference later in June, and do you have sort of any more update on the timeline regarding when we can expect to see the clinical study kick off?
No, there's no update at the moment. So we're basically, and I think we come back probably to that later also regarding the other programs. So we're still on time as indicated in the press release. Obviously, but also in this specific case, together with Transgene, have taken steps to minimize the risk of any potential impact of COVID on the timelines. And I would say so far so good.
Okay, that's good to hear. Looking at the abstract, it seems like, at least in the abstract, I don't know if that's true for the entire preclinical program, but it seems like the single agent effect, so just using BT001, is quite good. And so I'm curious if you're going to be targeting showing single agent activity in the first studies, or whether you will go more straight at a combination together with PD-1 inhibitors.
Yeah. So you're referring to the preclinical data, and there you could clearly see that you have both single agent activity of BT001, which is of course the combination in the way of the oncolytic virus and the anti-CTLA-4 antibody. But also we have very good activity when we combine it with anti-PD-1. And for the clinical trial, we'll explore both single agent as well as the combination.
Okay. Do you feel that it's important to show single agent activity in terms of possibilities for out-licensing and so on, or is the sort of industry looking more towards the combination anyway?
I think it's always helpful, let's put it that way, that you can also demonstrate single agent activity, especially for the old class, because looking forward, if you think about cancer therapy, it will be all about combination anyway. So I think from my perspective, the combination is actually more important because, as we all know, there will be no one magic bullet that will just eradicate cancer forever. So it will be always about combination. But it's always helpful if you also have single agent activity, and also that you can demonstrate it. But from my perspective, I think the combination is even more important.
Okay, I know you're very excited about this project. You are conducting it in collaboration with Transgene, which means that sort of there's two companies paying for it, which could perhaps sort of give a little more financial firepower. But generally, when it comes to a project such as this, at what stage would you think that this would be suitable for out-licensing? Do you have any thoughts on that?
So obviously, just to remind everybody, and you mentioned also, so it's a 50-50 collaboration between us and Transgene. That means everybody is contributing 50%, but also everybody will participate accordingly whenever we have something to earn out of this program. And that's obviously a business question that concerns both companies. And this is always, I think, being discussed, so we have a clear strategy. But then, of course, it will be also a case-by-case decision in case there are opportunities to discuss partnering. And what I'm saying now, obviously, would not give any opinion regarding Transgene, but I would think from my personal and Bioinvent's perspective, as soon as we have demonstrated some proof of concept, I think it would be ready to partner. In any case, it would then entertain partnering discussions. And of course, you always partner at the time when you feel it's right, in that sense that you're also getting the right value in return.
Okay, that's good to hear. Turning to the TNFR2 project, and you had another little baby here called 1910, which is another antibody within the overall TNFR project. Fascinatingly, I would say, it seems now that you are confirming that 1808 is an antagonist towards TNFR2 and 1910 is an agonist, and in sort of other terms is that sort of one of the antibodies are actually inhibiting, you could say, the receptor, and the other one is stimulating it, is perhaps another way to put it. But even so, these different effects seem to have positive effects, both of them, which are not contradictory to each other. And my question is here, how do you see this in terms of a package for, a partner? Are these two antibodies possible to out-license, individually, or do you see the entire TNFR2 project as a single project with several interesting antibodies, but probably of interest to one partner?
Yeah, I would guess, obviously it's still early days. And first of all, I think I use this also as an example, as I'm always trying to explain. So we have the integrated discovery engine, which is generating ample and multiple opportunities for clinical development. So I think that's a nice proof. And we never really come up with only one antibody to a target. We always would have a panel that we can look at and then consider for preclinical development and further clinical development. Looking at those two specific antibodies, and again, it's early days, and there will be some more details once the poster is presented at AACR. Currently, obviously, 1808 is clearly the front runner because there we also have done the toxicology, etc., etc. So this is clearly ready for clinical development, and as we also indicated, so we are ready to file a clinical trial application, IND, during this quarter, and we're still pushing that target. And 1910 obviously is a little bit later. And at the end, it depends on the partnering discussion. So from a development perspective, they are clearly distinct because you have to develop them as distinct compounds. And then theoretically, at least, you could also think to out-license or try to partner them to different parties. But obviously, you can also look at it from a package perspective that you say, okay, those are two anti-TNF receptor 2 antibodies. And you can, of course, also think about making a larger package. So that really depends on data, I would say, and on the partner or potential partner that you're discussing with. So at the end, it's very flexible. And I would say from a development perspective, those are two distinct compounds, clearly.
Okay, so for instance, you're not planning to use them, both in sequence or anything like that, where you could, like you showed with the sort of 4-1BB antibody, that you could actually, have different mechanisms in sequence, but that's not what you're planning here. So you're planning within the TNFR to, you could say, sub-projects that could be individually valuable to a partner.
Absolutely. And obviously, it's still, as I said, it's early days. So we're currently, developing 1808. That's where we are preparing for filing the CTA this quarter. And 1910 is not at the same level yet. So there we still need to do some more work. And there will be also some more details disclosed once the data is published in detail at AACR.
Okay. All right. So let's turn and ask a few questions about financing and the financing situation as well. I mean, when you present, you usually you list the different sources of financing that are available to Bioinvent. I mean, you have milestones from Pfizer, you have partnering events, you have the factory, you have raising money in the capital markets and so on. And with the COVID-19 situation, I mean, my guess would be that sort of partnering situations and milestones and so on, I mean, they're certainly not going to come faster anyway. And so, I mean, how do you see the sort of COVID-19 situation affecting you in terms of your financial flexibility?
I would say, I have to differentiate there a little bit. I would say regarding the flexibility, it's not affecting us at all. Regarding timing, obviously things will take a little bit more time. And there I also have to differentiate a little bit since, we are lucky as Bioinvent that we're in Sweden. So the company is completely 100% operative, which is very, very helpful. And that's why we can keep all our timelines that we have promised to the market. When it comes to the manufacturing business, to partnering, to ongoing collaborations, you of course never know how this will be affected, and there might be some delays at some time point. But we are not in a press situation, so we have still available finance, and I think later you come back to that question again when we talk about the Keytruda study, and maybe to address that already here. So we'll start as planned. There's no delay, and we feel confident that we can finance this study. So that's the only thing that I can say. So it will be, again, those 4 sources that you have mentioned: our manufacturing business, our collaborations, and maybe later we discuss a little bit about Pfizer, our partnering, and then, of course, also financing. And I think I mentioned in some earlier interview that I gave to Redeye. So luckily also we have very, very strong institutional investors on board. So I think I mentioned specifically Van Herk, but obviously we also have others. So I'm to put it that way, I'm not concerned about the financial situation of the company.
Okay. And I just wanted to go back there a second, Martin, and clarify my question I sent to you in advance, because I think it's quite important. So for the viewers, I had asked the question whether you thought it was possible to start the combination study with 1206 and Keytruda before a financing event, as normally, a company will want to be confident that they have the financing on hand already before starting a study to be sort of certain from an ethical perspective that they can finish it and the patients that are exposed, that the data is collected in the end and so on. So that was the question I asked, and you answered that you feel that you can start this study, basically when it's ready to start, and it's not contingent on a financing event as such. That was right, correct?
That's correct. We are comfortable to start the study.
Good. And basically you answered the other question as well. And Jan and I, we note that you have a mandate to issue about sort of 100 million shares, and we sort of think that, maybe the sooner the better, I think, because everybody's waiting for it. But I'm sure you're sort of planning that in a prudent way. So let's go on then and ask a little bit about the different studies in BI 1206, which is of course, really important. And we wanted to ask that, we've been waiting a while now to see a first patient in release from you regarding the 1206 Keytruda study. It seems a little bit slow to start, and again, we wanted to know, is this COVID related, or is there any other reason for it, being a bit sluggish getting off the ground?
No, actually, I think according to, and I just checked before we connected, the latest presentation that we have on our webpage. So I think we're on track, absolutely. And obviously, when you go back into last year, I think we had the IND filed quite some time ago until we then had finally the agreement with Merck in the box. And that was actually the main delay last year. We finalized the agreement at the end of the year. But since then, the target of enrolling the first patient is still on track and holds for Q2 this year. So we're on track.
Okay, that's good.
And we believe, maybe to give a little bit more color even, I think it's very likely that it also will enroll quickly, because there's quite some excitement of the sites which are involved.
Okay. And there are quite a lot of US centers, right? I don't remember. I looked at the number, the centers on clinical trials. I don't remember the exact proportion, but I guess.
Yeah. So the site. Yeah. Go ahead. Sorry.
Yeah, but I saw some pretty major US cancer centers on there. So I guess those are the centers you're referring to, right?
Exactly. So this trial will be run in Sweden and in the US.
Yeah. Okay, and then switching over to the combination study between 1206 and rituximab, and basically the same question. I know at one point you had hoped to have the expansion part of the study, going before the end of last year, and now it seems to be stretching out a little bit. Is this sort of, do you have any comments on that? And do you in any way fear that, if it takes a little time here, that, the clinical landscape changes in any way, so the design of the clinical trials could become, less relevant or anything like that?
No, I absolutely don't think so. So probably you remember that we had this very interesting ASH publication where we could show that 1206 plus rituximab was showing nice activity in all types of resistance to a number of different drugs, venetoclax, lenalidomide, ibrutinib. So the clinicians, and especially one name I would like to mention there, Michael Wang from MD Anderson, who is, you know, the, if you want to say it like this, the godfather of mantle cell lymphoma internationally. They're very excited about the more tolerable treatment that we have there for, potentially have there for late stage patients. Because those patients obviously just have a few or very few options left. And as you remember, so this opens really a quick path to registration. And just to remind you, so we have orphan designation for mantle cell lymphoma, which of course is also quite important in the context of the KOL principal investigator that I just mentioned, Michael Wang.
Okay, and I also just wanted to ask you, I asked you at, I think your Capital Markets Day some way back, if you felt that 1206 could, you know, be split between indications when it comes to combine with either rituximab or, for instance, Keytruda, and your answer was quite emphatically, no, that's probably not a good idea. I just wanted to check back and, you know, see whether that's still the case, and whether in that case, I mean, the biggest opportunity, at least the way I and Janne see it, is of course in the combination with Keytruda. So in a business development perspective, you know, it's not like you're going to have the rituximab data and go out and sell the drug, because that's not where the, you know, big money is going to be. It's going to be when you start to get data from the Keytruda trial, and therefore the timing of the rituximab combination is actually, I mean, from a stock market perspective, it's always good to have the data, but from your perspective in Bioinvent and the partnering possibilities, it may not matter as much. Is that correct, or how do you see that?
Yeah, absolutely. So I think the way I look at it, the non-Hodgkin lymphoma could be a very quick way to market. And I think it just adds to the application potential, and we didn't touch at all on autoimmune, because there could be also a potential application of 1206. But obviously, if you look at the markets, non-Hodgkin lymphoma is a much smaller market than potentially the solid cancers of the patients who do not respond anymore to PD-1, PD-L1. So absolutely, but I think as the way I see it, it probably will play out just right. And basically the first signs of clinical efficacy that we have seen and that we also have press released, I think is a very good pace maker in a way for the other study as well, because obviously the solid cancer study centers, they are aware about the data that we're generating in the non-Hodgkin lymphoma trial, and that's of course stimulating them as well. So I think at the end it will go nicely hand in hand. That's how I see it. Okay,
so you also see the rituximab study facilitating recruitment into the Keytruda trial. That's what you're saying basically, right?
Yeah, so basically it helps that we already have their first signs of efficacy, obviously, because people can see, okay, this thing is potentially doing something. Plus, obviously, we have also the dosing data such that we can start the second trial at higher doses already, such that we also have a quicker chance to see potential signs of efficacy in the second study.
How, I mean, you should be, do you feel that you are in the sort of highest dose, in the highest dose level right now in the combination with rituximab? Is it, I think you mentioned that you were right now at 100% saturation and seeing some good, you know, obviously you are seeing some good data. Do you feel it makes sense to go much higher, or are you close to the final dose, do you think?
We are close. So we have, with these 75 mg, we have 100% receptor occupancy, but it's what we would like to see is 100% receptor occupancy for a longer time. And I think what is also important, and I think we try to make it clear in the press release, is that the first signs of efficacy that we see are at suboptimal doses, because at 75 we see 100% receptor occupancy. The other signals we saw at 30 or 35, whatever it was. And so we were very keen actually to move a little bit higher, but we're close since we see now the 100% saturation, but we want to extend the 100% resector PNC a little bit longer. And then we also would expect to see even stronger signals and good data.
Okay, sounds promising. So, and also then I have to ask you the question I almost always ask you, and you usually answer, Mats, you're the only one who cares. And that is when can you give us any hint on when the sort of preclinical data from the PD-1 receptor and 1206 combination will be ready for publishing or presented at a conference or anything like that? I've been dying to see this data for about 2 years now, so I mean it better arrive soon, or I'm just gonna I don't know.
Well, put it that way. So obviously, Merck has given us Keytruda, and that's based on that data. And what we're trying to do is really to get that into a really high impact journal, and you know how that goes because they always ask for more than you have ready at the time. So from a corporate development perspective, it doesn't matter because we have made the decision to put it into the clinical trial. We are on time, and hopefully we'll generate soon interesting clinical data. That's the most important thing from a corporate value development perspective. And the publication, this is, in my view, this is just an add-on. And I think what I can tell you and the market, obviously, so that was part of the due diligence that Merck has done in order to come on board. And you can see that those type of deals in the past, they happened quite frequent, not so anymore. So they're very, very selective. And so that should give the market the confidence that the data is very good. Otherwise, you wouldn't get Keytruda for free. And the data will be published when we will publish it. So once we really have secured a high impact journal.
Okay, so Martin, let's then just round off with what do you feel that we have neglected or forgotten to ask you about that you would sort of like to make sure that we don't forget?
Yeah, I think probably covered everything. So from my perspective, it's just important that everybody understands that we're really on track, that we're really delivering, and not only this year, also last year, since now quite some time. And hopefully the markets will reward us at some time point for this. I think it's now looking a little bit better, but I think at least the Swedish investors are still looking and are still a little bit careful watching us, which is fine. Because if you look at all the different deliverables, we are exactly on track and so far have completely ticked off as promised since not only this year but also last year, and I think that's something I think that everybody should understand.
Okay, Martin, thank you so much for your time. We want to be sort of mindful and let you get back to work, and thank you so much for agreeing to come on this little shareholders show that we do, Janne and I. We really appreciate it, and so good luck with everything, and thank you again.
Thank you.
Yeah, thank you very much for your time, and hopefully we talk again soon.
Sure.
Love that.
Okay.
Thank you. Bye-bye.
Bye.
Thank you. Bye-bye.
Bye.